Good afternoon, everyone. It's my privilege. Usually the dean introduces our recipients of the dean's lectures. I'm Greg Kirk. I'm the vice dean for research. In Ellen's absence, I'm very honored to be able to welcome everyone, both online as well as in person, to today's dean's lecture. As most of you know, the Dean's Lecture Series is a chance to hear firsthand from our newly appointed or promoted professors. Today, we have the distinct pleasure of listening and learning from Scott Bailey, professor in the Department of Biochemistry and Molecular Biology. Scott's originally from Sheffield, England, following bachelor's and doctoral degrees in biochemistry and biophysics, respectively, at the University of Sheffield. Scott spent several years at Yale as a Howard Hughes Medical Institute postdoctoral fellow under the mentorship of Nobel laureate Dr. Thomas Stites. Since joining the Bloomberg School faculty as an assistant professor in 2008, Scott has led pioneering research employing innovative molecular and microscopy tools to better understand the structure and function of the cell's molecular mechanisms and how they modify DNA and RNA. When he first came to the school, Scott focused on what was then a relatively niche topic, the bacterial adaptive immune system called CRISPR. Scott was doing CRISPR work before CRISPR was recognizable in the lay public or referenced on TV shows. Today, CRISPR is far from niche. It's the basis of a growing number of molecular biology tools used across biomedical research, opening up revolutionary research and therapeutic opportunities. I'm going to leave it to Scott to get into the details, but very broadly, CRISPR is a bacterial defense system that can selectively cut specific sequences of DNA and is now being repurposed as a gene editing tool, among other applications. CRISPR can function essentially as a pair of targeted microscopic scissors with vast potential. CRISPR-based tools can likely help us to protect the microbes we use to produce food or pharmaceuticals, and also help to target human pathogens. More broadly, CRISPR approaches are rapidly advancing research on everything from cancer to infectious diseases. But to continue developing these tools, we need a better understanding of the CRISPR system itself. That's where Scott and colleagues come in. Scott is known for his innovative, thoughtful approach to this complex field, and he has consistently employed novel techniques and approaches. He received our school's Shikani El-Hibri Prize for Discovery and Innovation, as well as the university's President's Frontier Award, among other honors. He's helped our university to harness the power of cryo-EM technology. Cryo-EM is an advanced imaging technique that allows researchers to create high-resolution 3D maps of complex molecular structures, which have been cryogenically processed, in some cases down to the atomic level. It opens an incredible window to the molecules that make up the cells. As Scott was shifting to the promise of cryo-EM in his own lab, he played an instrumental role in establishing the Beckman Center for Cryo-EM here at Hopkins. He continues to support this facility and the community associated with it, but has also been sought out by individuals across the university for help with diverse cryo-EM issues and analyses. This is just one example of how Scott is not only a leading innovator and researcher, but also a team player who contributes beyond just his own lab. After spearheading BMB's PhD program and the admissions process for years, he recently became the PhD program director. Scott is known for being an exceptional mentor who brings a very personalized approach to working with each mentee in the lab, supporting their individual needs and goals. He's very committed to educating students in the classroom as well, carrying one of the larger teaching loads in his department and regularly receiving excellence in teaching honors. In fact, he's received that honor 12 times since 2020. In addition, Scott regularly steps up to support the broader mission of Johns Hopkins, answering the call to serve on school and university committees. Scott, we are very grateful to all that you do, all that you give to your department, to our school, to our university, and to your continuing contributions to the world of molecular biology. Thanks for all that you do and for sharing your work with us today. Thank you, Scott.
Okay, thank you for that great introduction, Greg. And I'm going to dive straight into it here. So today I'm going to talk about the molecular basis of bacterial immunity. But first I'm going to kind of correct this slide and sort of say, although I'm kind of a spokesperson for this work and the person who stands up here and gets kind of the comments about me, in reality this is what we call BLAB, so the Bailey Lab, which, sorry, fingers and thumbs. So these are all the people that have been through my lab, and like a relatively recent picture of everyone who's been through my lab since I started at Hopkins. So mainly PhD students and master's students, but really, this is how the sausage gets made. I do this bit, but all the actual work I'm presenting comes from these guys. And so that's, I'm going to start with that kind of acknowledgement. We'll come back at the end. I will also start acknowledging the funders, partly because of the times we're in, but partly because I do get money from Intellia Therapeutics. And so some projects are sponsored by Intellia, so I should sort of disclose that before I carry on. I've also had generous funding from the Walder Foundation, which was a big help in kind of spearheading and letting us be able to move into cryo-EM. It's actually invaluable in that regard to get to cryo-EM. Okay, so back to the molecular basis of bacterial immunity. So what I'm really talking about is CRISPR, which Greg sort of introduced. And this kind of dystopian kind of esque picture here, this is a bacteriophage infecting a bacterial cell. And although, like many people well, everyone I guess would be aware that CRISPR is used for genome editing and is a tool used in molecular biology, it didn't evolve to do that. Obviously, it evolved to protect bacteria and archaea, or prokaryotes, from infection by bacteriophage. So bacteria, just like us, are like bacteriophage are the viruses that infect bacteria, and bacteria under constant pressure from viruses. So this is this micrograph here is actually an electron micrograph. This is Chesapeake Bay water. And so if you look at, I don't know if you can quite see it, the big sort of fingerprinty smudges, those are bacteria. And then all the little kind of like black blobs that are around, those are bacteriophage. And so in most environments, bacteriophage outnumber bacteria by about kind of 10 to 1 is like a reasonable estimate. And you put that into context, human body, 10 to the 13 cells. It's probably a similar number of bacterial cells that hang around in our body, maybe up to 10-fold more, but probably about a similar number. But it's about 10 to the 16 bacteriophage particles in each person on average, mainly on the skin and in the gut. And so bacteriophage, they infect bacteria, they carry genes into bacteria, they're involved into the natural transformation of bacteria. And so they're kind of a dark matter of biology in the sense that they shape bacterial populations. And obviously bacteria have huge health burdens for human population. So bacteriophage, we're interested just in the basic biology because of this basic dynamic between bacteria and bacteriophage. So the other thing about immune systems and what we've kind of already alluded to is that study of immune systems has been an incredibly rich sort of avenue for development of tools and therapies. So this is not an exhaustive list, but maybe a big list of we have sort of antibodies, which obviously we use every day in sort of like at the bench in sort of Western blots, but also a lot of therapies are now based on antibodies. RNAi, again, is used at the bench and in the clinic. Restriction enzymes have been used, not so much as a therapeutic, but it's certainly a revolutionized molecular biology. And then more recently, CRISPR-Cas9 has been harnessed as a genetic tool and as for gene editing. And I just highlight in here that the antibodies in RNAi are basically eukaryotic, and the restriction enzymes in CRISPR-Cas9 are prokaryotic in origin. So studying prokaryotic immune systems has been very beneficial. And there's been Nobel Prizes for the discovery and application of each of these systems. So I'm going to choose this. And one thing I'm going to point out now is that the two of these, the RNAi and the CRISPR, are driven by base pairing. And it's base pairing that I'm going to kind of focus on today in the talk. And so what I mean by this is obviously just nucleic acid base pairing. So the Chargraff's rule. So as seen by Watson and Crick, GCAT. So if you have two strands of nucleic acid with a DNA or RNA, they will come together and base pair. And this provides exquisite specificity. We can make depending on the length of these things, but we get exquisite specificity from this and affinity. And it's these two immune systems, RNAi. And CRISPR take advantage of this base pairing. It's one of the reasons why they really exploded, just because of the simplicity of that mechanism. So the problem that we face when we're using CRISPR as a tool is it's not what CRISPR evolved to do. So it's tuned to a very specific role. And so we can think about balancing specificity with promiscuity. So if you're targeting a phage viruses, they evolve very quickly. The genomes are dynamic and change as much as they can. They're pretty plastic. And they do that because they're evading immune responses. And so we have this balance, but if you want to make a tool, you want that to really be balanced towards specificity. You want your tool to be as specific as possible. If you're going to put Cas9 in a patient, you need it to only cut at the site it's programmed to cut. And so that's critically important. But that's not how it's evolved to do. It's evolved really to be more promiscuous than that. It needs a certain level of specificity because it needs to target the phage and not, say, the host chromosome. But it's really much more promiscuous than we would want from a tool development standpoint. And this limits what we call phage escape, which is the ability of a phage to just avoid avoid immunity. So that's kind of a backdrop of where we kind of come into this. And a note on CRISPR systems is that they're very, very diverse. And this is kind of a calling card of immune systems. Because viruses evolve very quickly, it leads to an arms race with immune systems. So immune systems also evolve very quickly because viruses very quickly evolve mechanisms to circumvent the immune system. And so I'm not going to belabor or go into a lot of detail on this, but there are essentially two classes of CRISPR system, class 1, class 2. Class 2 is the Cas9 system, where class is a type 2 system, so that would be Cas9. It's a single protein, introduces double-strand breaks into DNA. The class 1, which we're going to talk about today, are much larger multi-subunit complexes. Their RNA base pairing is much more is much longer. The RNA it uses to guide, and it's more diverse. There are 4 subtypes within this. So, just to introduce the fact that we're going to talk about a system that's actually a little bit distinct from Cas9. Class 1 systems are being used as tools. They're a little bit less tractable than Cas9, but they offer many advantages. The RNA is longer. They tend to be more specific. For using these in natural systems, where you can take advantage of a system that's been expressed, the type 1 systems and class 1 systems are actually the most abundant CRISPR systems in nature. So if you find a CRISPR system in nature, it's more often than not a class 1 system, but the class 2 are actually quite rare. So we'll just look a little bit more specifically at type 1. So type 1, 2-component system. So we have a Cas3 protein, which is a helicase nuclease. Its job is to actually destroy the DNA. It's the nuclease that destroys DNA. And we have a complex, which we call cascade, multiple subunits, and it houses the CRISPR RNA. So in some sense, this is kind of a targeting part or arm of this immune system. It's responsible for recognizing the threat, and then Cas3 is responsible for destroying the threat. And we can just step through that a little bit. So this complex will search for a target sequence. So for those that sort of know, there's PAMs. I'm not going to talk about PAM sequences much today at all. I'm going to concentrate on base pairing with a protospacer, but I'll highlight it periodically. But in order for the cascade complex to recognize a target, it has to have a protospacer, which is a region of the DNA that's complementary to this RNA that's in the complex. And it has to have an adjacent PAM, which is a small motif, usually on the order of 2 to 6 base pairs. But like I said, I'm not going to talk too much about that. Once it's paired, in this particular system, by pairing you've got double-stranded DNA that has to pair to RNA, and so one strand of the DNA is released as the other strand pairs with the DNA. sorry, whereas one strand pairs with a CRISPR RNA, the other strand is released, the other strand of DNA is released, and that's where Cas3 gets recruited, and then Cas3 degrades the DNA. So this is a system where it's not a precise cut like Cas9, it actually degrades the DNA and chews it up. And actually, if you compare these systems as immune systems, type 1 systems are much more robust immune systems, probably because they just chew up the DNA and aren't just making these cuts that in principle could be repaired. So in sort of coming into this, so like the first kind of hit we had was maybe like about just over 10 years ago now. The fundamental question we had is we know that we have a guide RNA, which we short to CRISPR RNA, and this light green region in here is the region that corresponds to the DNA target. We have a DNA target, it has this small motif, this PAM, and it has a protospacer, which is the region that base pairs to the guide. And so the real question was, well, how does this happen? How do you open up a DNA and how do you read that sequence? And that's kind of where we came into this, is to try and understand this problem. And so initially, this was kind of before kind of EM kind of revolution took off. And so at the time, we were reliant on X-ray crystallography. So like, you know, we really wanted to understand how Cascade facilitates this base pairing. And so crystallography, just to kind of summarize, we grow a crystal, literally a three-dimensional building block of a complex. We shine X-rays at it, we get a diffraction pattern, and from that we can calculate a map, build a model, and then we've got a sort of an atomic view of what's going on. And the idea is kind of form is function. If we know how it looks, we can really start to understand how it works. And so this process is slow. Each of these steps takes real time. And it took us about 5 years from having this complex made to having a structure for us to be able to do this. And I'm just highlighting that here, because when we get to EM, we'll see that that's now much, much quicker. And you're also limited with what will grow in a crystal. Like you crystallize these complex molecules, which in of itself is amazing. These things actually will crystallize and form these regular lattices of these complex molecules. But you only kind of get what the crystal gods give you in a sense of what will crystallize. And so although we were trying to get crystals of a complex bound to double-stranded DNA, we actually only got crystals bound to single-stranded DNA. But that answered the first question for us. So we solved this structure, and here's the structure. So it's a large, complex, 405 kilodaltons. It's likened to, it looks like a seahorse, where you have sort of head up here, the body, and down to the tail. It has a bunch of subunits. In there, there's 5 different genes and the CRISPR RNA, and they're they form around this CRISPR RNA to form this large complex. And so, so when we look at this complex, we start to think about, this base pairing. So the first thing I want to do is look at the RNA. And so I'm just going to fade it to the RNA. And the first thing you notice is that this is the guide RNA in the CRISPR complex. And this is kind of what you'd expect it to have looked like. This is what we would call sort of A-form RNA, the typical kind of spiral. It's a single strand, but that's kind of what you would expect. If you look at the structure of Cas9, this is what you would see. But in Cascade, it looks very, very different. So if we look at that a little bit more closely, so we bring in the DNA, and we see that it's still base pairs, there's base pairing occurring. We can already see that there's kind of strangeness happening because there's holes in here where we don't see that in the A-form here, which is, again, what we'd expect to see. So look at that a little bit more closely, and what we see is that the two strands aren't wrapping around each other. So this is, there's 32 potential base pairs across this orange strand here, and so you'd expect it to have wrapped around itself 3 times, just based on canonical kind of what the structure of DNA is, or RNA-DNA hybrid, but it doesn't wrap at all once. Okay, so it's flattened out, it's unwound essentially. And so you can see maybe more clearly when we put the space fill on there. So what's going on here? So we can look at this a little bit more closely. And what you see is that every 6 base pairs actually flipped out. So basically, as there would normally be a base pair if you were thinking about just a regular base pair. And what actually happens is one of the strands goes down and one of the strands goes up. And it flips it out, and you get this hole in the middle. So you're isolating segments of 5 base pairs. When you have a flipped out base, you get 5 base pairs, and you get a flipped out base. So we can modify our original schematic to look like this. And it turns out that when I talked about class 1 complexes, so now we zoom forward, about 10 years ago, we got the first structure. You zoom forward to now, and every class 1 structure looks like this. They all look like this. No matter what they do, what the biology is, they all look like this. So it's a common kind of scaffold for this process. Okay, so if we go back to the RNA, and we can look at like, well, why is it like this? It's like this because of the proteins. And so we can bring in some of the proteins. I'm just going to talk about a few of them here. So we have a protein that caps one end of the RNA, Cas6, and then we hit these Cas7s, and the Cas7 is the one that actually is distorting that nucleic acid. So if we rotate around so we can look at it, this is the structure of Cas7. And all structural biologists like to sort of liken various structures to various everyday things. So this we liken to a right hand. So it has fingers domain, palm domain, and thumb domain. The RNA drapes over this, so between the fingers and palm, and you see that the thumb kind of sticks out there. And if we zoom into the thumb, what we can see is the thumb is what's causing that base to flip out. So when you put the DNA back in and you look, you see that the thumb is occupying, but there's not really a hole, there's protein there. So remember from a very first cartoon, Cascade's pre-assembled with the RNA and searches for the DNA. So the RNA structure here would all be there first, and then as it binds the DNA would pair, and that RNA base would have been flipped out, so there's nothing to pair to. And so it would do the first segment, skip over, do the next segment as it pairs. So that's why it looks like this. And we think the reason probably this evolved is that the RNAs are so long, you have 3 turns. That would be very difficult if you think about unwinding DNA and then wrapping 3 turns around the single strand of DNA when you reveal it. So we think it's a topological problem. So this doesn't happen in Cas9, but Cas9's RNAs are much, much shorter. So I think that's probably, it's difficult to prove this kind of thing, but we think that's probably one reason why it looks like this. So we can go back to the structure, and then we can just fill it out. You can see the Cas7s, the thumb domain actually reaches over to the next subunit, and multiple Cas7s kind of filament across the guide region. Then we can go back to the big complex. And then the Cas7s go all the way to the end, and then the end is capped by another subunit, which we'll not talk about today. And then the remaining subunits, which were involved in sort of DNA and PAM recognition, fill out the complex to the final complex. And then it was at this time, sort of roughly, that kind of EM came on the scene, and Hopkins got the scope, or were able to saw that getting a scope of Hopkins. So EM has a very different kind of workflow. This is a microscopy technique. So we basically take our sample, we freeze it on a grid, we put it on a electron microscope. We literally just get projection images of all the particles. And then we do a bunch of things in a computer to basically reconstitute a 3-dimensional map of that model. The thing that for us, there's lots of, you can compare X-ray crystallography and EM, and there's lots of kind of pluses and minuses. The thing for this project, and for a lot of the projects in my lab, which were the reason why EM is a big sort of step forward, is it's significantly faster. So from going from the first structure of Cascade that we did, from being able to make the complex, I told you it was 5 years before we had a model and we were able to publish the paper. Now when we take a new complex, similar complex, and we just make it, it usually takes us less than 6 months to have the structure. Obviously it's science, so sometimes it's longer, but it can, we've several times now we've been able to solve similar structures very, very much quicker. Nearly orders of magnitude quicker. And the other thing is that we can, when we put images on the grid, we're no longer constrained by a crystal, and we can see multiple states of the protein. So if a protein is existing in different conformations, we can deconvolute that and see that when we do this technique, which we can't with typically with X-ray crystallography. So we get kind of like dynamics in the sense, we're not measuring dynamics because we don't have a time component, but we can sort of approximate dynamics when we look at this. And so we switched then at this point to using EM to determine structures of Cascade, to further look at these mechanisms. And now we can use double-stranded DNA. And we get structures, we can develop these structures, and this particular one I'm showing is 2.4 angstrom resolution. So the X-ray structures were actually at low 3s. So in this case, with a molecule of this size, we actually get better data and better maps from EM than we do X-ray. It's not always that way around. It depends on the size of the molecule and how its sort of properties in ice and things. But these produce much, much better maps. And one thing we were able to do when we did this is that we were able to see different states. And so we see the state that we wanted, or we kind of aimed for, which is the RNA base paired to DNA. And we see that, the strand here is base paired all the way across. We don't see the non-target strand because it's flexible. And if it's flexible, we don't see it in maps. But it's in the map, it's just moving. But we also see intermediate states. And one of the intermediate states we saw was one where we just captured the initial binding event between the complex and the target DNA, where we'd recognize the double-stranded DNA and the PAM, but we'd also formed the first two base pairs. And so this was, although this had been suspected from a lot of biochemical data over the years, it kind of cemented this idea and let us visualize this idea that base pairing starts next to the PAM and then proceeds in a wave over the protospacer. So, you know, we don't know this for sure, but it's very, it's easy to think about this, that because of that flip type base, that things would occur in these segments, and that we would see that you would see that there'd be a stability to having a given segment fully base paired, then the next, then the next, then the next, then the next. And so basically what we're getting at here is when we think about, well, how does Cascade read sequences? It basically reads sequences exactly as you do. It starts at one end and just reads it across. And so to kind of illustrate that point, if we just kind of think about how Cascade is working. So here's what do you call these things? Like a puzzle or whatever to look at. So here's just full alphabet, and you can try and find the word elephant, okay? So you're gonna do that, essentially how Cascade does it. Yeah, you're gonna start looking for that EL, and then look for an E, and you're gonna read across, and probably most of you will have found elephant on this one. But Cascade is sort of more impressive than that because it's doing this with a simplified alphabet. Now, unfortunately, simplified alphabet makes this kind of word search problem harder. So then if we go to this one, so now we've simplified it down. It's not even quite as simple. I think there's 5 letters in elephant, but it's a simpler alphabet, and now it's a little bit harder to find elephant, yeah, because you've got that simplified alphabet. I can't tell if you guys are finding it or not, but you can find it. But again, there's 81 bases on here. We're reading 81 bases at a time, so it's very, very simple. So let's make it a bit bigger. And so now I get $20 when anyone can find elephant. You'll not find it'll be harder to find it, but it's on here. This elephant is on here, and it's up there. I'm doing it quickly, so I don't have to pay $20. But again, this is still a very small number of bases, and even if I fill the screen so you can't read it, it's still not enough bases to even approach how many bases are in a bacterial genome or in a typical bacterial cell. This room would be filled with bases for that to occur. And so it's doing this at incredible speeds and incredible precision when it does it. But the take-home message here is that it is doing it exactly like you do it. Okay, and so then we sort of turned with this. We got this notion of how it interacts with double-stranded DNA. I want to keep this kind of relatively straightforward story. And so we zoomed forward sort of 6 or 7 years now in terms of research in my own lab on this problem. And we've looked at things like DNA binding, the recognition of a PAM, this kind of region. That's very important for the biology. We've done stuff on that. But like I said, I'm going to keep it to base pairing just to keep it the sort of message simple here. And what we think about then is like, well, what if you're reading this long word here, and you start over here and you go all the way across, then what if there's an error? What if the DNA has one base missing, an extra base, or has a different base, but it's not complementary? How many errors have to be in there before it doesn't bind? Is it one? Is it, what is the deal? And so there's been a lot of biochemistry done by my lab and other labs to kind of think about this and analyze this. And I'm not going to show any biochemistry. I'm going to stick to molecular stuff today. But what we call the seed region, essentially those first 2 segments, is you have very little tolerance for any changes in that region. And we think that's because that's where base pairing starts. So at that point, if you've only found, say, 5 base pairs, if there's an error in 5 base pairs, like percentage-wise, you think the energetics are binding, then that's going to be a bigger penalty. because you've just only, you're only looking at 5 base pairs. So if you have errors over here, you really interfere with binding of cascade, and you get no immunity in cells. But as you make more and more base pairs, you're going to see that, when you get over here, you've already formed 15 base pairs when you're into this like 4th segment over here. So just energetically, you've just got less of a problem if you think about the kinetics of binding and that binding process we discussed. And this bears out if you look at sequence. If you analyze sequences and you do this in cells, do it in vitro, you see that what we call this PAM distal region, just sort of half away from a PAM, you can see that you can tolerate changes in this region. And in this region, if there are changes, you still get binding, and you still get immunity. So from a standpoint of a tool, this is bad news. These are off targets. And so what we wanted to understand is, like, how does an off target work? So the off target is just the complex binding to a site that's not a perfect match to the RNA. Happens with Cas9, happens with Cascade, and maybe less so with Cascade just because of the length of the RNA. But yeah, and so this is like, we wanted to understand this from a molecular perspective. There's been a lot of work on the genetics of this, kinetics and thermodynamics of it, but we wanted to understand sort of structurally was what was missing. And so that was kind of our next kind of phase. And now with EM, we can do this because we can do lots more structures much more easily. We're not doing, it's not taking us 5 years to solve a structure. Now, it would have been quicker to do X-ray than 5 years per structure at this point, but it still would have been very slow, and the maps are not as good. So if we think about this, there are actually 12 possible, I'm going to start with mismatches. There are 12 possible mismatches between the RNA and DNA molecule. So we've got the kind of Watson-Crick, the Chargaff rules of like AT, CG, and then the opposite, GC, UA for RNA. But if you think about mismatches, you can have purine-purine mismatches, pyrimidine-pyrimidine mismatches, and you can get purine-pyrimidine mismatches if you don't have the sort of Watson-Crick pairs. So there are 12 theoretical, or not theoretical, there are 12 mismatch possibilities. And so what we did is we designed a whole bunch of targets. And I'm just showing, this is just one kind of library we used. This has got 6 off targets in it, OT is off target. And what I'm showing here, the sort of color-coded thing at the top, that's the CRISPR RNA. It's sequence. And every 6 base pair is grayed out because it doesn't form any base pairs. And then we have the on-target sequence, which is anti-parallel, drawn, so it's like it's base paired, and it's all grayed out. And then what I do is I draw out these off targets, and I just color when there's a mismatch. And so you can see where the mismatch. So we've put the mismatches over here, which is where the PAM would be over here. So this is the PAM distal region, and all these targets function as off targets. So if you had a virus, or you have you can use this with plasmids, but if you have a piece of DNA that has these sequences, all these sequences behave exactly the same. They all behave like the fully complementary on target. Okay, so they're genuine off targets, despite the fact that each has got 3 mismatches. So in total, we did 9 of these off targets, and we sampled each potential mismatch at least twice, some up to sort of, I think, 4 or 5 times. And so we end up with this, we solve the 9 structures plus 2 structures with the on target. And then we got, what is that? So it's like we got nearly 30 different base pair mismatch structures from this screen. And so I'm not going to show you all, I'm not going to wade through all these things. I'm just going to show you some examples and then give you the rules that we kind of saw. So the examples here are here. So I'll just explain these figures. So in the sort of ghostly white thing in the background, that's the Watson-Crick base pair in the on-target structure. So that's what it should look like. And then, or not should, should's probably the wrong word, but that's what it looks like on the on-target structure. And then the colored base pairs, those are what we see in the mismatch. Okay, so in each case, the green one is the RNA and the orange one is the DNA. So the green one, you see, like is barely moving, and then the orange one is moving around a lot. So you see that the sequence changes. So the white one here would have been a C, but it's really a G, and even here we get sort of a spinning round of a base to a syn position. So it can actually form what is called a Hookstein base pair. We get helix widening over here. We get rearrangements. This is just a wobble base pair, and we get rearrangements here to form base pairs. And so, like I said, I'm not going to wander through all these because there's a lot and it's boring if I just go through them all like that. But big picture, what are we seeing? as we look all at this. And, this is a lot of work to go through all these kind of structures. This took many months to kind of wade through all these structures. And I should say the resolution of these maps are 2.2 to 2.4. So we can say with pretty high precision what's going on here in these things. And our sort of take home on this was that the CRISPR RNA is very rigid. It basically doesn't move. It's just, it's sort of literally a guide. It doesn't move, it doesn't distort. This is very different from Cas9. Similar studies have been done on Cas9, and Cas9, the RNA actually accommodates a lot of the distortions. But in Cascade, it's rigidly held by those Cas7 subunits, and it basically refuses to move. The DNA absorbs all the distortions. And in fact, the protein has no role in this. That DNA strand that base pairs to the RNA basically has no contacts with protein. And there's no new contacts with any of these distortions. So it's all nucleic acid based that absorbs it. So that's actually a little bit of a bummer, because what you were thinking going into this study is, could we find sites in the protein we could mutate to make Cascade more specific? Because if we could mutate Cascade to discourage off-target binding, to not accommodate these mismatches, that would make a more specific enzyme. But that actually turns out to be almost impossible based on the mechanism at use, because it just doesn't use the protein at all. It just lets the DNA absorb it all. And then if we look at the various types of mismatch, so purine-purine mismatches, we have that flipping of one base. So we get a Hookestein base pair, or we get helix widening. The helix is not restrained to be sort of standard A-form width, because the DNA can move out. Pyrimidine-pyrimidine mismatches just make any adjustment they can to try and form a hydrogen bond. So like if you have a CC mismatch, they kind of collapse in a little bit, change the angle of the bases and just form one hydrogen bond. So they do what they can. In some cases, they just don't form hydrogen bonds and they just sit with a gap. And then purine pyrimidines, they just shift as much as they can to form maximize hydrogen bonding. So this would be the classic wobble base pair. So we see kind of like wobble-like base pairs in the structure. So that give us a view of how mismatches are accommodated, and why Cascade can hold on to these mismatches. And I should say that here we're viewing the final product once it's bound. So the mismatches would likely look the same if they're in the seed region. The problem there is one of energetics and sort of kinetics, that these base pairs, although these distorted base pairs form, it's harder for these to form than a Watson-Crick base, but you pay more energy to form these. So that's why when it's in that seed region, when you're trying to initiate base pairing, that's why that fails and you don't see binding. Because these would be, you got to flip bases, you got to widen grooves, you got to do stuff to distort. But once you've got enough binding energy, these can settle into these conformations. Okay, so the other form of mismatch that we've looked at is what happens if you have an extra base or a missing base. So this actually there was no data for biochemically or even genetically, for various reasons. But so the first thing we did, I'm not going to show the biochemistry today, but the first thing we did is we did use libraries to basically look what happens if you delete a base in the DNA at each position all the way across the protospacer. And then, so you've got an RNA that's one base longer than the DNA. And the position is just changing. And we did the same thing with insertions. And we inserted all 4 bases and went all the way across and inserted it so the DNA had an extra base in it. We do that, and basically what we see is a similar pattern. So just broadly speaking, a similar pattern to mismatches is that the seed region is not as tolerant as the PAM-distal region. The PAM-distal region is really tolerant to these kind of changes. And so, again, we're interested in, well, how is that, how does it do that? How is that possible? And so we did EM of a few of these structures. I'm just going to show you one example of each, because broadly speaking, the mechanisms are the same for the different forms of this. And so first one is a deletion. So this is, we've just removed a single base. And what we see is that we get what we call base skipping. So the RNA strand looks, same deal. RNA is green. White is the what would be on target. Green is the off target structure. And so we look at this structure and between the two, the RNA doesn't move. The RNA stays the same. But the DNA distorts heavily. The DNA's got a missing T in this case. It's centered right in the middle here. And what happens is the bases just kind of squeeze over. They just stretch over to skip this base. So this is kind of a spaceful representation. You see here it's a bit wider and there's an extra base that's not paired. And this is actually the most distortion we see in any off-target, because you can look at down here, all these base pairs, the DNA does move around quite a bit. So it's the most distorting from a structural kind of perspective. But this is how it accommodates it. And again, we have this kind of like appearing rule that the RNA just does not move. And then if we look at an insertion, what we see is that the insertion, it's always kind of remarkable that nucleic acid can do this, but it does it time and time again, is that it just literally flips that base out, and it's able to form almost undistorted base pairing, or basically undistorted base pairing before and after the flip out. And that's just, this occurs, you see this in DNA repair pathways. If you look at complex RNA structures, you see this kind of pattern. The backbone is just able to flip and accommodate that insertion, easy by just flipping it out like that and maintaining base pairing. But again, we've got this idea that the RNA stays exactly the same. The DNA is what absorbs that base extrusion. Absorbs by base extrusion the extra base. And again, this is different from Cas9. In Cas9, at least with deletions, I don't think there is an insertion structure, but in Cas9, the RNA can do the base skipping rather than the DNA, but we don't see that in Cascade. I'm going to kind of bring this all together with kind of a movie here, which kind of just sort of summarizes where we're kind of at. Is this going to start on its own? There we go. Okay, so we're starting with the CRISPR RNA. The colors are a little different, unfortunately. And then this is not like a molecular assembly pathway. This is just Disney. It's just everything just kind of coming together, forming the complex. So that's the complex we've been looking at of Cascade. And then we were thinking about how it works now. So it's scanning the DNA. It will look for PAM. We've not discussed that in any detail, but it will look for PAM, and it'll start looking for base pairing. If it doesn't find base pairing, it'll keep moving. Particularly, again, if there's any mismatches in that seed region. But when it finds a seed region that matches, and again, we draw it in this kind of very orchestrated way of doing a segment at a time. We don't know if that's true, but it's gratifying. And then it will move across. Base pair all the way across, and in there could be those mismatches, there could be deletions, and then it recruits Cas3, and then Cas3 can lock on and can start to degrade the DNA, and that's kind of a molecular basis of the immunity. And so that's where we are right now. That there's in terms of kind of a future for this particular project, there's lots of things. There's lots of, we now understand these base pairing rules. We've got a lot of information about PAMs. There are other proteins that are involved in this system that help the system generate new RNAs. Because at phage, although we've talked about how the system is designed to be relatively promiscuous in that PAM distal region, and so it can tolerate mismatches, phage still come up with ways to sort of circumvent this system. They can mutate that PAM, they can mutate residues inside the seed, and then the system fails. And so what the system has done is it's evolved a process where it makes new guide RNAs that are a perfect match to the phage that's invading. And so the next sort of phase for us is to kind of investigate that side of things for this project. Okay. So I'm going to go back to the acknowledgement slide, and I do have one. I guess if we've learned anything from Marvel movies, is that you need a post-credit sort of teaser. So I do have a post-credit teaser when I've just gone through the acknowledgements here, which I'll show in a minute. So again, all of this, the project I showed is a subset of the people in here have worked on it, but none of this happens without lab and without all the people in lab. As I said, this is how the sausage gets made. And so I'll take questions in a minute and I'll show that extra slide. But what I'm going to do, and I'm going to embarrass them all, is I'm going to get everyone in lab. So if your name's on this slide, stand up. And I'm going to finish, because I'm not taking the applause on my own, because I didn't do this on my own. These are the guys that did it. It's always good to embarrass them. And so my post-credit thing is we have a lot of stuff. We have other structure. We have, I talked about the diversity of CRISPR systems, a lot of diversity. We've done structures of all the different sort of diverse systems. We have a lot of that coming with EM at the minute. But the other area my lab is kind of moving into, or has been moving into for a while, but we started getting there, is methyl reading in the context of a nucleosome. So it's a very different project, it's very different stuff, but this is why it's a teaser. So the methyl status of a DNA, whether DNA has a methylation on CPGs or not on the C, is vitally important to many processes in transcription in human cells, in our cells. And this is regulated by proteins that sense this methylation. And so we are now getting to the point where we can use EM to look at this. And this is, our main motivation comes from this in the context of cancer, but it actually impacts lots of different diseases as well. But two main players that we're looking at is UHRF1 and then NBD2. And so hopefully in the near future, we will be producing structures or papers describing these new structures. Okay, with that, I will take questions.
Very nice that you acknowledged the people in the room as well as those who've moved on. We are opening it up for questions. My first question, you would expect someone who is so focused on structures to have good graphics and videos, but those are impressive. Who does those? Is that someone on that list, or are you doing those yourselves?
Everyone except the one with the black background. I think I made all of them. People may have made them, some of them, but I think I made all of them.
All right, very impressive graphics. Let's open it up for questions. And those of you online, if you would like to, show your face and ask a question in person, that would be great. We also can follow questions on the chat. We got a question in the back.
Hi, Scott. Fantastic presentation. I have two questions. One, you would imagine, you mentioned at the outset that the type 1 system is much more widely present than the type 2, while the type 2 seems to be a much more simpler system in terms of the number of components. And so evolutionarily speaking, you would have imagined it would be the other way around, naively thinking. So what are the advantages of this type 1 system versus the type 2? And also the second question is, there are many species, bacterial species, that have multiple CRISPR systems. And so how do they coordinate that process? What is used? And so how does this differentiate there?
Yeah, so the first question, as far as we understand from doing sort of culture experiments, where you can grow different cultures, or you can grow a single culture that contains bacterial populations that have different immune systems, and kind of look at who survives longest from phage infection, and you can sort of play the games of looking at fitness costs of having a CRISPR system. The type 1 systems come at a much higher fitness cost, which is, I think, what you were getting at with what you'd think the Cas9 system would be better. But the Cas9 system is just not as good an immune system. So it's just not as strong as the immune system, because you're basically introducing a double-strand break, which is repairable. Now, bacteria aren't as good at double-strand break repair, typically most bacteria, than we are, but it still is enough to not give you a super sort of immunity, or this level of immunity compared with type 1 systems, where you really just chew up the DNA. It's more difficult for it to repair. So as far as we know, type 1 systems have proliferated more just because they are better immune systems. And it's not a little bit more, it's a lot more. Like it's like 90% of bacterial cells that have a CRISPR system have a class 1, not a class 2. So it's a big skew. And I've already forgot. Sorry, what was the second question?
Competition between multiple CRISPR systems.
Oh, yeah. So that is a little bit more enigmatic. We don't really know. The best kind of like educated guess is that the different immune systems are tuned probably to different phage, or like some phage may have developed methods to circumvent one type of immune system, CRISPR immune system, but not another. So that's why, bacteria may harbor multiple CRISPR systems. In terms of the interplay, It depends. So the class 1 systems, a lot of times the gene, the CRISPR array itself, which transcribes the guide RNAs, the same sort of guide gene can provide CRISPR RNA for all the type 1 systems in the cell. And so we've seen that's been well established. You can't do that with class 2 versus class 1. The RNAs aren't compatible. They're different fundamentally in terms of sequences. And so they would have to have a separate gene. So their interplay between is not well understood at this point. Yeah.
Great. Other questions? Yeah.
Beautiful talk, Scott, as always. So just curious, this base pairing structure, the intermittent base pairing structure, every 6 nucleotide flipping out. It's very fascinating. Do you know if it occurs in other R loops? Where this promiscuous kind of a binding could allow for some type of regulation in bacteria or even in mammalian cells.
As far as we know, it's unique to CRISPR systems. And only CRISPR systems
It's
do
only
that.
specific for Cascade. Because
For class 1, so all class 1 systems do it. So class 1 systems are quite diverse. Some only work on RNA, but they all do that. They all have the same structure and do that flipping out because they all have that Cas7 gene, which does that.
yeah, why wouldn't mammalian cells, where hundreds of thousands R loops could potentially form, where this kind of a
Yeah, I think in a eukaryotic cell, there's not the same like the R loops are often not intended. Yeah, like it's transcription. You get them in transcription from like errors in transcription, and so they're just naked nucleic acid. They're not necessarily proteinated, and it's the protein that drives that sort of crazy structure. So I think that's probably why. I mean, certainly, maybe there's something like that, and we've just not found it yet. But to my knowledge, no R loop in a eukaryotic system is really been shown to have that particular. And Cas7 is unique to prokaryotes. There's no equivalent gene.
All right. Hey, Scott. Amazing talk. I was curious about thinking about the CRISPR RNA being rigid. My first question is is the cascade complex what's making it so rigid? I imagine all these proteins holding it very tightly. And then the other question is there any insights into does the CRISPR RNA have a limit? I don't know, does it ever disintegrate from the cascade complex, or does it get degraded after a certain point?
I see. So you're right. It is the proteins that hold it's that Cas7 interaction is so tight. That's why the RNA can't move, because it's just completely surrounded by the Cas7 protein, and it sort of has kind of it basically has kind of intimate contact with it apart from the bases, which are displayed out to recognize the DNA. So that's why we think the RNA doesn't move, because it really can't. It's kind of really bound in there. As far as we know, once the RNA is inside Cascade, we as far as we know, it's sort of stable in there. It doesn't get destroyed. We don't really, one thing I didn't really go into, but when Cascade binds the DNA and Cas3 gets recruited and starts chewing away, it only goes in one direction, typically, in class 1 systems. It moves away. And Cascade is then stuck to DNA. We don't know how it gets recycled, even if it gets recycled. It's just stuck to the DNA, and nothing that we know removes it. So when we do experiments in the lab, it basically just stays there. So we don't really understand how that gets broken down. So maybe there is a mechanism where the cascade gets recycled in some way, but no one's ever seen that. And if you do Northern blots and things like that, once cascade is formed, those RNAs are super stable.
Hey, Scott, very nice talk. I was wondering, given the level of promiscuity, what's the likelihood that the bacteria would target its own DNA? And if that's high enough, does there have to be mechanisms that limit that in some way?
Yeah, so I didn't go into that today, but the PAM is what's controlling that. So the PAM is necessary. One of the reasons PAM is necessary is because it's not in the CRISPR array. So it's not in the gene that makes the RNA. So that, in principle, stops self-targeting, at least at the CRISPR array site, where you've got a perfect complementary match to that RNA. Other sites, I mean, at some level, you have to get into kind of a single-cell organism mantra that, like, it doesn't matter if some cells self-target and die, because other cells won't do it. And what we know is if you measure the rate, you can look at the kind of rates of it. Typically, fastly replicating DNA is targeted more efficiently than none. We don't really understand why, but that's the empirical observation. And so that's one of the reasons why we bias phage DNA, or not we, but why phage DNA is biased. But I think it's built as much as possible to only target phage, but if it does target self and that cell dies, then in a single cell kind of mantra, other cells will not do that and they'll survive. That's good enough.
You mentioned earlier about the promiscuity, and I know there are labs using CRISPR to target unique mutations, for example. And but I'm curious what you think the limitations are there in terms of given the promiscuity of the sequence target.
Yeah, so it's what I think what typically happens, particularly when you get sort of therapeutic and you really like start talking to people who are doing this therapeutically, is, you know, you have a choice on like the species, like which CRISPR system you use, which Cas9 you use. There are Cas9 mutants that are in principle more specific. So you can test those. You can test usually different guides. You can move around where you're binding. I mean, if it's a cancer mutation, you're trying to like hit exactly on the cancer mutation, that's more difficult. But you usually have got some sliders to where you can put it. And so what they typically do is just brute force through all those things. And the good news about this is it's easy to measure off-target activity. So at least in like tissue culture. So it's very simple to do, like, you know, the high throughput sequencing, very sensitive methods that can measure off target. So we typically know or can predict at least what's going to happen. Obviously, when you move into an animal or a human, things can change because that's not tissue culture. But at least we can test it, and that usually holds up as they do move through systems. So it's usually a game of screening rather than sort of rationalization right now. But theoretically, you know, Cas9 with 20 base pairs should be enough for any unique site in a human chromosome. It's not repetitive in any way.
Yes.
Thank you, Dr. Bailey, for the very insightful talk. I was curious to know, when does the Cas7 protein know when to stop reading the DNA sequence? And at which point does it activate Cas3 to degrade the complement DNA?
So Cas7 filaments on the RNA. And so there's only as many Cas7s as there are, as the length of the RNA. So it's a multiple of 6. So 1 Cas7 per 6 RNA. So every multiple of 6, you get a new Cas7. And then in terms of like when that stops recognizing, it's not so much the Cas7, it's the RNA. So when you get to the end of a guide and the last base pair is base paired, that's when it happens. That's when that stops. And then in terms of Cas3 recruitment, I didn't go over that, but there's actually a subtle conformational change in cascade. And so when base pairing gets towards the end is when that conformational change happens, and that licenses recruitment of 3. So 3 is only recruited if you form sort of base pairing from one end to the other, is the bottom line. So you know you've got full. You can have mismatches and things in between, obviously, but you've got basically all the way across.
Please forgive me if this is basic. This is a new subject for me, but I was wondering what the frequency of the PAM sequences are in the genome and if it's uniformly distributed.
No, it's a good question. They're very common. So like a Cas9 is a GG. So GG, I mean, we could do the math, but I can't do math on demand. But we could do the math of how many GG, but it's pretty common to have a GG with only 4 bases. There is, genomes are not, it's not a random distribution. There's obviously bias and things in there. And people have looked to see if there's been a bias in like the genome to sort of exclude PAMs, but there hasn't been as far as we can tell because they're just too small a sequence. So they are very frequent. E. coli's chromosome, which is all this system is from, will have many, many, many GGs. But in terms of searching for a phage with a GG, that matters from an immune perspective, it has to have a guide sequence after it. So when Cascade is searching the genome, it's searching for those GGs. Well, Cascade is not searching for GGs, it's a different sequence, but it's searching for those PAM sequences. So although there's a lot of them, it's a lot less than the whole chromosome or the whole genome inside a cell. So it speeds up the search process from that standpoint. But yeah, it's sort of a necessary evil to sort of, for this system to have evolved, basically, to work.
Make it quick.
You stressed on this PAM sequence, which is absolutely necessary. Raised this question of how the GG is recognized by the Cas9. I'm not sure if I.
Yeah, so PAM recognition is through proteins. So in Cas9, there's 2 arginines that actually hydrogen bond to the CC. That's opposite. That's how that molecular recognition occurs. Cascade, it's a little bit more complicated because it's a little bit bigger and it's more promiscuous for PAM. Recognizes more sequences than just one. But yeah, it's protein-protein contact, nothing to do with base pairing.
All right. And with that, we'll have to consider that our last question. The great thing about this school is how the diverse range of topics that are discussed in our hallways. And it's been great to see many of you from different departments asking questions. So I want to thank everyone who participated, both online and here in person. Certainly to get promoted to be a professor at Hopkins is not for the weak of heart. It's a long and arduous path. I echo the thanks for many of the people on the slide and in the room, but let's once again give a hand to Scott. Congratulations. Thanks very much.